Hi, hello students. Welcome to Mass Physics MNR. This is MRK Zoology faculty. So, in the previous class, we are discussed about what is menstrual cycle, including menstrual cycle. We are clearly discussed some problems like some multiple choice questions. We are clearly discussed, and along with the menstrual cycle, we are discussed about gametes, male and female gamete structural information. And up to the last class, that is over. See, in this today class we will discuss human reproduction, sixth class in human reproductive system. This is the sixth class, and this class is maybe last, it is. So in this sixth class we will discuss fertilization and implantation, and pregnancy and embryonic development in human being we will discuss today. Okay, so up to last 5 classes వరకు మనం menstrual cycle కంప్లీట్ అయింది. ఈ మెన్స్ట్రల్ సైకిల్లో మనం కొన్ని మల్టిపుల్ చాయిస్ క్వశ్చన్స్, మెన్స్ట్రల్ సైకిల్లో కొన్ని ప్రైమరీ ఊసెట్ సంబంధించి కొన్ని క్వశ్చన్స్ మనం క్లియర్గా డిస్కస్ చేసినాం. ఓకే, సో వాటితో పాటు మనం గ్యామెట్స్, స్పర్మ్ అండ్ ఓవమ్ స్ట్రక్చర్ని మనం క్లియర్గా డిస్కస్ చేసినాం. దట్ ఈస్ క్లియర్. ఓకే, నౌ we will discuss what is that fertilization and implantation, and pregnancy and embryonic development. So we will discuss today, and maximum today we'll complete this chapter in this sixth class. Okay, right, come to the point. First here, fertilization and implantation. So this is the today topic, fertilization and implantation. Fertilization and implantation. Right. See, fertilization means what? The process of fusion of two different gametes is called fertilization. So the process of two different gametes means here one is the sperm and second one is the ova. Okay, see, and okay, within the human female, within the fallopian tube, especially within the fallopian tube in ampullary region, these two different gametes are get fused and form the zygote. That is clear. Okay, so before this we'll discuss during the intercourse, where does the ejaculation takes place? After completion of the ejaculation, how the sperm is enter into the uterus from the cervix, and at the time of single ejaculation, how many spermatozoa are released into the female reproductive tract as a semen? Okay, we will discuss that. Before the starting this fertilization, first we'll discuss the ejaculation. Ejaculation means the process of releasing of semen into the female reproductive tract. Okay, the process of releasing of semen releasing is called ejaculation. Where does the ejaculation takes place? Means సెమెన్ ఏ పార్ట్ ఆఫ్ ద ఫిమేల్ రిప్రొడక్టివ్ సిస్టమ్ లో రిలీజ్ అవుతుంది అనేది మనకు తెలియాలి. సెమెన్ రిలీజింగ్ ఎజాకులేషన్ టేక్స్ ప్లేస్ ఇన్ ద వైజైనల్ రీజియన్. విదిన్ ద వైజైనా ejaculation takes place. Okay, within the vagina ejaculation takes place. It is clear. See, at the single time, at the single ejaculation, within the single ejaculation, how many spermatozoa are there in the semen? Okay, in a single ejaculation, in a single ejaculation approximately 200 to 300 million sperms are 200 to 300 million sperms spermatozoa are released, or spermatozoas are there. Okay, it is the first point. Say in these 200 to 300 million spermatozoa, how many spermatozoa are present in active condition? How many spermatozoa are present in inactive condition? At the same time, how much percentage of sperms are having normal shape and structure, and how many spermatozoa does not have normal shape and size or structure? Okay, see, ejaculation means releasing of semen into the female reproductive tract, especially that is the vagina. It is clear. In a single ejaculation, approximately 200 to 300 million spermatozoa are released. This is one point. Okay. See, for example, in the ejaculated semen, approximately 200 million sperms are released. 200 million sperms are there. For example, okay, see these 200 million sperms, when we are considered as 100 percentage, in this 100 percentage of sperm, 40 percentage of sperm do not have what is normal shape and structure, do not have what is shape and size, do not have shape and structure, shape and structure, shape and structure, do not have shape and structure, and the remaining 60 percentage of sperm having what is have normal shape and structure, normal shape and structure. It is one important point. Normal shape and structure. It is clear. Okay. Now, when we are considered these 60% equal to now again 100 percentage. These 60% again we will consider as a 100 percentage. In this 100 percentage of what is which sperms are having normal shape and structure, here again 40% sperms are having 40 percentage of sperms have what is vigorous motility. Vigorous motility is there. Vigorous motility is there, and remaining 60 percentage of sperm do not have what is motility. Do not have motility. Do not have motility means motile capacity, means which are not having a what is swimming locomotion capacity. Even the presence of normal shape and structure, which are not able to swim inside the female reproductive tract. Okay, so this is one important point. Means after ejaculation, how much percentage of sperms are able to enter into the uterus from the vaginal through the cervix region? Means only 40 percentage of sperms are having ability to enter into the uterus through the cervix region from the vagina region. Okay, very clear. So I'll draw the one little diagram for this. So it is the female reproductive system. Okay, it is the what is fallopian tubes, and it is the uterus for easy understanding. Okay, and here this is the what is vagina region, and it is the vaginal canal. It is the vaginal canal. It is vaginal canal, and here this is the endometrium. It is one. Next side, it is the endometrium, right? And it is the uterine fundus. Uterine fundus, right? See, it is the vagina. Vagina. This is the cervix. Cervix. It is the womb, means uterus. Okay. Where does the ejaculation takes place? The ejaculation takes place within the vagina region. Here the ejaculation takes place. Now this is the semen. It is the semen. Okay, so ejaculation here. Okay, so within the ejaculated semen, for example, 200 million sperms are there. Okay, in this 200 million, how many spermatozoas are able to enter into the uterus fluid means, okay, how much percentage of sperms are having normal shape and structure? In this total number of spermatozoas, only 40% of sperms are not in the total ejaculated semen. The total how much percentage of sperms are having normal shape and structure? In this percentage of sperm, only 40% of sperms are able to enter into the, okay, able to enter into the where uterus fluid, because which are having a motility capacity. ఇప్పుడు ఈ స్పర్మ్ కి స్విమ్మింగ్ కెపాసిటీ ఉంటది కాబట్టి ఇది సెర్విక్స్ ని క్రాస్ చేసి డైరెక్ట్ గా యుటరస్ లోకి వెళ్తుంది. ఎంత పర్సెంటేజ్ స్పర్మ్స్ వెళ్తుంది అంటే, ఎంత పర్సెంటేజ్ అయితే టోటల్ ఎంత ఉంది ఇక్కడ, అది నార్మల్ షేప్ అండ్ సైజ్ ఎంత ఉన్నాయో దీంట్లో 40%. సో ఇక్కడ ఇవి ఎజాకులేట్ లోనికి స్పర్మ్ లోకోమోషన్ జరుగుద్ది అన్నట్టు. ఇది ఫర్ ఎగ్జాంపుల్ మీకు ఒక ఈజీ అర్థం కావడానికి ఒక క్వశ్చన్ ఇస్తాను. ఫర్ ఎగ్జాంపుల్, in a ejaculated semen, 1000 spermatozoas are there. In a ejaculated semen, 1000 sperms are there. Here the semen is ejaculated. Here only 1000 sperms are there. Okay. In this 1000 sperm, okay, this 1000 sperm we are considered as 100%. In this 100%, okay, 40% of sperms do not have normal shape and structure. Do not have normal shape means this is first word, do not have normal shape and size. In this 1000, 40% is 1, 60% is 1. Okay, in this 1000, 40% means here 400, 60% means here 600. It is clear. Means these 40% of sperms not having shape and structure, and 600 sperms are having normal shape and size. Okay, now this 60% means this 600. In this 600, like it is in this 60%, now this 60% it is called as, means these 600 sperms now we are considered 100%. మళ్ళీ ఈ 600 sperms ని 100% గా కన్సిడర్ చేసుకుందాం. ఇలా కన్సిడర్ చేస్తే ఈ 100% లో, ఓకే, 40% sperms are having vigorous motility. ఓకే, విగారస్ మొటిలిటీ అనేది వీటిలో ఉంటది. అంటే విగారస్ మొటైల్ కెపాసిటీ, విగారస్ మొటైల్, అండ్ రిమైనింగ్ 60% లో మొటిలిటీ ఆబ్సెంట్. మొటిలిటీ ఆబ్సెంట్ హియర్, మొటిలిటీ ఆబ్సెంట్. ఇట్ ఈస్ వెరీ క్లియర్. అంటే ఈ 600 లో 40% ఏంటంటే అప్రాక్సిమేట్లీ 250 sperms are having vigorous motility, and remaining 360, okay, 240 sperms are having more vigorous motility, and 360 sperms are not having motility capacity, even the presence of normal shape and size. Okay, only means in the 100, in these 1000 spermatozoas, only 240 sperms are enter into the cross the cervix region with the help of what is the sperms are having a flagellar tail. With the help of tail, only 250 sperms are enter into the uterus fluid. Remaining how many? Approximately 360 plus 400. So these are present outside the vagina. Means these are present outside the cervix or out of uterus. So 360 plus 400, approximately how many? 400. Here 760 sperms are present. What is in these 760? 400 sperms are not having normal shape and size, and even the presence of normal shape and structure, 360 sperms are not having the motility capacity. Okay, so ila motility capacity లేనివి, నెక్స్ట్ నార్మల్ షేప్ అండ్ స్ట్రక్చర్ లేనివి, ఇవన్నీ కలిస్తే 760 స్పర్మ్స్ అనేవి యుటరస్ లోకి వెళ్ళే కెపాసిటీ ఉండదు. ఓన్లీ ఈ 1000 లో 240 స్పర్మ్స్ మాత్రమే యూటరస్ ఫ్లూయిడ్ లోకి వెళ్ళి ఇవి ఫాలోపియన్ ట్యూబ్ లోకి రీచ్ అవుతాయి అన్నట్టు. సో ఇది ఇప్పుడు మనకి ఇక్కడ డిస్కస్ చేసిన 200 మిలియన్స్ ఆఫ్ స్పర్మ్స్ లో కూడా 40% అసలు నార్మల్ షేప్ అండ్ సైజ్ ఉండనే ఉండదు. సో ఇక్కడ 60% కు నార్మల్ షేప్ అండ్ స్ట్రక్చర్ ఉంటది. కానీ దీంట్లో ఈ 60% మనం 100% కన్సిడర్ చేస్తే ఈ 100% లో 40% స్పర్మ్స్ కేమో విగారస్ మొటిలిటీ ఉంటది. అంటే ఆ విగారస్ మొటిలిటీ ఉన్న స్పర్మ్సే అవుట్ సైడ్ ఎజాకులేటెడ్ ఏరియా నుంచి యూటరస్ లోకి వెళ్తాయి. అంటే ఇక్కడ సెర్విక్స్ ని క్రాస్ చేయాలంటే మొటిలిటీ కెపాసిటీ అవసరం. సో ఈ సెర్విక్స్ ని క్రాస్ చేయాలంటే విగారస్ మొటిలిటీ ఉన్న స్పర్మ్స్ మాత్రమే సెర్విక్స్ నుంచి యూటరస్ ఫ్లూయిడ్ లోకి వెళ్తాయి. అదర్వైజ్ వెళ్ళలేవు. సో దీంట్లో 40% మాత్రమే, ఈ 100% లో 40% మాత్రమే యూటరస్ ఫ్లూయిడ్ లోకి వెళ్తాయి. రిమైనింగ్ 60% నార్మల్ షేప్ సైజ్ ఉంటది. కానీ వీటికి మోటిలిటీ కెపాసిటీ ఉండదు. మోటిలిటీ కెపాసిటీ ఉండకపోవడం వలన ఇవి ఇక్కడనే ఆగిపోయి ఇక్కడనే చనిపోతాయి. ఇక్కడనే స్పర్మ్స్ డై అవుతుంది. సో ఇట్లా ఎవ్రీ total how much percentage is ejaculated in these ejaculated semen. So how much percentage of sperms are having normal shape and structure? In that only 40% of sperms. Okay, so not in the total released sperms 40%. So which sperms are having normal shape and structure? In that sperms only 40% is able to enter into the what is spermatozoa, what is uterus fluid. So this is very clear information, very clear information it is. Please follow this. Okay, so there is no doubt here, very clear information. This is the what is semen ejaculation process. See, within the semen, already we are discussed in previous class that contribution of semen. The semen contain what is large amount of fructose, calcium ions, and certain enzymes are there. In these certain enzymes, some kind of substances are prostaglandins. Okay, some kind of prostaglandins are there. That prostaglandins are helps to movement of this cervix. Actually, in a normal time, generally the normal condition, the cervix is present in tightly closed condition. Okay, so when the cervix become open, means by the action of the presence of prostaglandins within the cervix, within the vaginal region. So how the prostaglandins are reached here? Along with the ejaculated semen. ఇప్పుడు సెమెన్ లో ప్రోస్టాగ్లాండిన్స్ ఉంటాయి. ఆ ప్రోస్టాగ్లాండిన్స్ ను సర్విక్స్ ను స్టిమ్యులేట్ చేయడం వల్ల సర్విక్స్ ఈస్ గివ్ ద లిటిల్ స్పేస్ ఫర్ మూవ్మెంట్ ఆఫ్ స్పర్మ్. ఇది లిటిల్ స్పేస్ ఇవ్వడం వల్ల ఆ స్పేస్ లో స్పర్మ్స్ అనేవి స్విమ్ చేసుకుంటూ యూటరస్ ఫ్లూయిడ్ లోకి వచ్చేస్తాయి. వన్స్ యూటరస్ ఫ్లూయిడ్ లోకి రీచ్ అయిన తర్వాత యూటరస్ ఫ్లూయిడ్ లో ఉండే ఫ్లూయిడ్ లో స్విమ్ చేసుకుంటూ బోత్ ఫాలోపియన్ ట్యూబ్ లోకి రీచ్ అవుతాయి. ఇప్పుడు లెఫ్ట్ ఫాలోపియన్ ట్యూబ్ లో సెకండరీ సైట్ ఉంటే ఆ లెఫ్ట్ ఫాలోపియన్ ట్యూబ్ సైడ్ వెళ్ళిన స్పర్మ్స్ వాటితో ఫెర్టిలైజ్ జరుగుద్ది. అండ్ రైట్ ఫాలోపియన్ ట్యూబ్ లో ఉంటే రైట్ సైడ్ వెళ్ళిన స్పర్మ్ తోటి ఓవము ఫెర్టిలైజేషన్ జరుగుద్ది అక్కడ. దిస్ ఈస్ ద what is ejaculation process. Okay, right. What happened after ejaculation we are clearly discussed. Okay, after that here, right, after ejaculation come to the fertilization process. After ejaculation come to the fertilization. See, in this fertilization process, once again I'll draw the detailed neat diagram of the uterus. Okay, see, after ejaculation Okay, the 40% of sperms are enter. See, once the sperm enter into the what is uterus fluid, it is undergoes to first process, it is that is the capacitation. Okay, when it is enter into the what is uterus fluid, here capacitation process is done. What is the meaning for capacitation? Okay, the capacitation process done before reaching the fallopian tube. Okay, before reaching the fallopian tube or before reaching the secondary oocyte, capacitation process occur. See, capacitation means the process of removal of cholesterol. Okay, it means what? The process of removal of cholesterol and plasma membrane surrounding the spermatozoa, surrounding the sperm. Okay, that process is the capacitation before reaching. For example, it is the sperm. Okay, it is the sperm and it is the nucleus and it is the acrosome. Surface of the spermatozoa, plasma membrane and cholesterol is there. Plasma membrane and cholesterol is there. See, before reaching this sperm to the fallopian tube or to the secondary oocyte, what is this plasma membrane can be really removed. So now when it is reached to the what is secondary oocyte in the fallopian tube, and that without plasma membrane contains sperm is reached to the secondary oocyte for the fertilization. What is advantage the removing of this cholesterol and plasma membrane from the surface of the spermatozoa? That is the question. Okay, for example, if the plasma membrane is not removed from the surface of the sperm, what will happen? Okay, so ఇప్పుడు ఈ ప్లాస్మా మెంబ్రేన్ రిమూవ్ కాకపోతే ఏం జరుగుద్దంటే, ఫర్ ఎగ్జాంపుల్ సరౌండింగ్ ద ఎక్రోజోమ్ దిస్ ప్లాస్మా మెంబ్రేన్ ఈస్ దేర్. ఇప్పుడు ఎక్రోజోమ్ కు సరౌండింగ్ ప్లాస్మా మెంబ్రేన్ ఉంటుంది. ఇప్పుడు ఎక్రోజోమ్ ఫంక్షన్ ఏంటంటే, ఇది ఓవులార్ మెంబ్రెయిన్స్ ని డిజాల్వ్ చేసే ఎంజైమ్స్ ని రిలీజ్ చేస్తుంది. ఎక్రోజోమ్ హ్యాస్ ఏ వన్ మేజర్ రోల్. That role is releasing certain enzymes. That enzymes are used for dissolving the ovular membrane. Surrounding the secondary oocyte, how many membranes are there, or how many layers are there? 2 layers are there. Clear? This after dissolving these 2 layers, okay, the sperm nucleus enter into the ooplasm of the secondary oocyte. Otherwise, without dissolving that outside ovular membranes, the sperm nucleus is not able to release into the ooplasm of the secondary oocyte. It is very important point. Understood? So that's why whenever the enzymes are released, these enzymes are not move out from the plasma membrane. Then how can the ovular membranes can digest or can removed? Any chances are there? Means there is no chance. That's why this plasma membrane should remove from the surface of the spermatozoa. Should remove from the surface of the spermatozoa. So this is the capacitation. It is clear. It is the capacitation. After completion of the capacitation, without plasma membrane, the sperms are reached to the both fallopian tubes. For example, the spermatozoa is present here. I mean, secondary oocyte is present in the. Okay, the secondary oocyte. I'll draw the one little big picture, big diagram. So it is the fallopian tube. For example, it is the fallopian tube. These are the fimbriae. Okay, and here the secondary oocyte is there. It is a secondary oocyte. Surrounding the secondary oocyte, zona pellucida is present. At the same time, corona radiata also present. At the same time, corona radiata. This is the secondary oocyte. It is the secondary oocyte. Secondary oocyte is present. Now, after completion of the capacitation, the sperms are reached to the where what is fallopian tube, and these are all are reached to the ampulla. See, ampulla is the fertilization zone. In which place of the fallopian tube, okay, the sperm fuse with the secondary oocyte, that zone of the fallopian tube is called the fertilization zone. So what is the fertilization zone in fallopian tube? Means that is the ampulla. Ampulla is the fertilization zone. Now, when the sperms are reached to here, these all the sperms are surrounding to the, these all the sperms are get surrounding to the secondary oocyte. For example, in this 40% of sperm, okay, few million sperms are move towards right fallopian tube, and few million sperms are reached to the left fallopian tube. And to the sperms, they don't know what is in which fallopian tube secondary oocyte is there. Either maybe left fallopian tube or maybe right fallopian tube. So that's why we are move left fallopian tube, we are move right fallopian tube after completion of the capacitation. Okay, see, for example, the secondary oocyte is present in the left ovary, left fallopian tube. Okay, in that month, the left ovary is complete the ovulation. That's why the secondary oocyte is present in the left oviduct or left fallopian tube. Okay, so right whenever the what is these all the capacitation sperms are upper capacitation, the sperms are get surrounding to the secondary oocyte in the fallopian tube, means in the ampullary region. Okay, once the sperm reach to the surrounding the secondary oocyte, actually within the ampullary region, within the what is surrounding the what is secondary oocyte, one acidic medium is there. That acidic medium is the hyaluronic acid is present. Hyaluronic acid. Hyaluronic acid is present surrounding the secondary oocyte. What is advantage the presence of what is hyaluronic acid surrounding of the secondary oocyte? Okay, this hyaluronic acid is helps to protection of secondary oocyte from the pathogens. Okay, from the pathogens. Okay, from the pathogens here. So, for example, if any other pathogens are enter, if any other pathogens are enter, what is try to enter into the secondary oocyte or try to damaging the secondary oocyte, these hyaluronic acid get immobilize the pathogens. Thereby, the pathogens are not able to enter into the secondary oocyte, and secondary oocyte will not destroyed by the pathogens. Then how the sperms are get survived surrounding the secondary oocyte within the hyaluronic acid? Okay, here one reason is there. That reason, okay, that reason is what is these sperms are, okay, these sperms are can release hyaluronidase enzyme. Okay, the sperms are can release the hyaluronidase enzyme. Hyaluronidase enzyme. Hyaluronidase enzyme is released. Now, when these sperms are released the hyaluronidase enzyme, what is the hyaluronic acid can immobilize, means that is that acidic nature will be decreased. Okay, the acidic nature will be decreased. Understood? By the decreasing the acidic nature at these spermatozoas, these sperms are get more close to the what is secondary oocyte. These are contacted to the corona radiata, outer layer of the secondary oocyte. So this is the reason. Simple thing. ఇప్పుడు సెకండరీ oocyte చుట్టూ హైలురోనిక్ యాసిడ్ అనేది ఉంటుంది. మరి హైలురోనిక్ యాసిడ్ ఎట్లా రిలీజ్ అవుతుంది అంటే surrounding the secondary oocyte corona radiata cells are there. These corona radiata cells are releasing the hyaluronic acid, acidic substances. ఓకే, ఇప్పుడు ఈ హైలురోనిక్ యాసిడ్ ఉండడం వల్ల అదర్ ప్యాథోజన్స్ అనేవి ఈ సెకండరీ oocyte ని డ్యామేజ్ చేయడానికి డ్యామేజ్ చేయలేవు. రీజన్ ఏంటంటే ఏ ప్యాథోజన్ అయినా ఎసిడిక్ మీడియంలో సర్వైవ్ కాదు. కాబట్టి ఈ హైలురోనిక్ యాసిడ్ అనేది ఫస్ట్ ప్రొటెక్ట్ చేస్తుంది. ఇది ఫస్ట్ పాయింట్. ఓకేనా. నెక్స్ట్ మరి ఇప్పుడు ఈవెన్ స్పామ్స్ కూడా ఎసిడిక్ మీడియంలో సర్వైవ్ కావు. మరి ఎట్లా స్పామ్స్ అనేవి ఎసిడిక్ మీడియంని క్రాస్ చేసి కరోనా రేడియేటర్ని కాంటాక్ట్ కావాలి అంటే కరోనా రేడియేటర్ వరకు రీచ్ కావాలి అంటే ఈ స్పామ్ ఎక్రోజోమ్ ఉంది కదా, ఎక్రోజోమ్ నుంచి ఓకే, ఇట్ ఈస్ ఫ్రమ్ ఎక్రోజోమ్. ఓకే, ఎక్రోజోమ్ నుంచి వాట్ ఈస్ హైలురోనిక్ ఎంజైమ్ అనేది రిలీజ్ అవుద్ది. హైలురోనిక్ ఎంజైమ్ అనేది రిలీజ్ అవుద్ది. ఈ హైలురోనిక్ ఎంజైమ్ అనేది ఈ హైలురోనిక్ యాసిడ్ తో మిక్స్ అయినప్పుడు ఈ ఎసిడిక్ నేచర్ డిక్రీజ్ అయిపోద్ది. Means surrounding the secondary oocyte the acidic medium will be converted into basic medium or alkaline medium after mixing of these hyaluronidase enzyme surrounding the secondary oocyte. Thereby the sperms are easily more close to the secondary oocyte and which are all are contact to the corona radiata. Understood? ఇది ఫస్ట్ స్టెప్. డిక్రీజింగ్ ద ఈ ఫెర్టిలైజేషన్ లో ఫస్ట్ స్టెప్ ఏంటిదంటే వాట్ ఈస్ కెపాసిటేషన్ ఒకటి. సెకండ్ స్టెప్ డిక్రీజింగ్ ద ఎసిడిక్ మీడియం. ఓకే, సెకండ్ స్టెప్ ఏంటంటే డిక్రీజింగ్ ద ఎసిడిక్ మీడియం. ఈ 2 స్టెప్స్ ఉంటాయి. నెక్స్ట్ ఆఫ్టర్ దట్ ఎక్రోజోమల్ రియాక్షన్స్ ఆర్ స్టార్టెడ్. ఎక్రోజోమల్ రియాక్షన్స్ మీన్స్ ద ఎక్రోజోమల్ రియాక్షన్స్ ఆర్ ఇనిషియేట్ ద డిజాల్వింగ్ ద ఓవులార్ మెంబ్రేన్స్. What are corona radiata and zona pellucida layers can be destroyed here. So by the presence of particular secretions, these secretions are released from the acrosome. Okay, we will discuss that point with the next slide. See, for example, right here from the acrosome, what type of secretions are released? See, already we are clearly discussed the acrosome is modified which cell organelle. Means the acrosome is the modified Golgi complex cell organelle and which is able to produce a certain enzymes. Okay, first one hyaluronidase. Hyaluronidase enzyme. It is the first one, hyaluronidase enzyme. And the second one corona penetrating enzyme. Corona penetrating enzyme. Corona penetrating enzyme. And the third one is the acrosine. So these enzymes are released from the acrosome. See, what is the role of hyaluronidase? The hyaluronidase helps to decrease the acidic nature. Decrease the acidic nature. Decrease the acidic nature means hyaluronic acid acidic nature will be reduced. Next, the role of corona penetrating CPE. Okay, corona penetrating enzyme means it is dissolve the corona radiata. Dissolve the corona radiata. Dissolve the corona radiata. Okay, next role of acrosine. It is dissolve the zona pellucida. Dissolve the zona pellucida. Dissolve the zona pellucida. This is. So these 3 types of secretions has a 3 types of particular role for 3 different functions are done by these 3 types of secretions which are from the acrosome. This is the point here. Okay, right. Now this is the what is a secondary oocyte. Okay, and surrounding the secondary oocyte, what are present here? First, zona pellucida is present. It is a non-cellular membrane. After that, what is corona radiata? This layer is the second layer and cellular. Okay, for example, this is the sperm. Okay, this is the sperm. This sperm, what is it? Is the head and it is the acrosome. This acrosome is first continues to the corona penetrating enzymes releasing. Means it is continuous to the coronal reaction. So coronal reaction for really dissolving the corona radiata. Coronal reactions occurred for the dissolving the what is corona radiata. Okay, so that millions of all sperms, how many millions of sperms are surrounding to the single secondary oocyte? These all spermatozoas are releasing the corona penetrating enzymes, thereby the entire surrounding the secondary oocyte, the total corona radiata get dissolved. The total corona radiata get dissolved. See, this is the secondary oocyte. Surrounding the secondary oocyte, what is it? Is the zona pellucida. Okay. Now there is no corona radiata. Here what happened? Corona radiata get dissolved. Dissolve the corona radiata. Dissolve corona radiata. Dissolve corona radiata. Okay, it is clear. After that, without corona radiata, what is the zona pellucida and what is secondary oocyte is there. Now, this sperm is, and again it is contact to the, these all sperms are contact to the zona pellucida. Once the zona pellucida contact by these all the spermatozoas, now here these acrosome can release acrosin enzyme for zonal reaction. For zonal reaction. Okay, for zonal reaction. Understood? After that, what happened? See, surrounding of the zona pellucida, many spermatozoas are there. Many spermatozoas. It is clear. Many spermatozoas. Okay, but only one sperm acrosome can able to dissolving the zona pellucida. Once the zona pellucida get dissolved, okay, what happened? That sperm nucleus can be released into the secondary oocyte. For example, it is the secondary oocyte. Okay, it is the nucleus of the secondary oocyte. Now, when the sperm, what is, and this is the zona pellucida. It is the zona pellucida. In this region, what is the sperm nucleus can be released. From here, the sperm nucleus is released. The sperm nucleus released into the ooplasm of the secondary oocyte, and outside this, that empty, what is middle piece and tail. Middle piece and tail are present outside. Middle piece and tail are flagella. These are present outside the zona pellucida. So after releasing, now sperm nucleus released. Sperm nucleus released. Sperm nucleus released into ooplasm of the secondary oocyte. Ooplasm of the secondary oocyte. Ooplasm of the secondary oocyte. This is one. And after that, what happened here? Once the sperm nucleus is released, immediately the zona pellucida becomes rigid. Okay, now it is the secondary oocyte, and within the secondary oocyte, two nucleus are there. One is the secondary oocyte nucleus, second one is the sperm nucleus. These two are there. Again, what is the zona pellucida becomes more rigid. Zona pellucida becomes more rigid here. Zona pellucida becomes more rigid. It is very, very important. And once the zona pellucida becomes rigid, which cannot dissolve to the action of other spermatozoas. It means the zona pellucida can allow only one sperm maximum. Okay, so the entry of single sperm into the what is secondary oocyte, okay, that is called monospermic condition. If two sperms are enter into the secondary oocyte, bispermic condition. Okay, bispermic condition, monospermic condition. Maximum one sperm is allowed into the secondary oocyte. Okay, only one sperm is allowed into the secondary oocyte. That's why that is the monospermic condition. By the entry of single sperm, that is called monospermic condition. Monospermic condition. So one sperm, single sperm enter. Single sperm enter. And if two sperms are enter into the single secondary oocyte, bispermic nature. Okay, it is the, now this is the sperm nucleus. It is the sperm nucleus and it is the secondary oocyte nucleus. See, once the sperm enter into the, so when does the second meiosis will complete in the secondary oocyte nucleus? Means after entry of sperm into the secondary oocyte, the secondary oocyte will complete the meiosis 2. Okay, when where the nucleus complete the meiosis 2 here. The nucleus complete the meiosis 2. In this process, it is the secondary oocyte. I'll draw the little big diagram here. And this is the nucleus of the secondary oocyte. And this is the nucleus of the spermatozoa. White color is the nucleus of spermatozoa. It is. Now, once the sperm, see, what is the reason the secondary oocyte nucleus initiate the meiosis 2? Means, along with the nucleus of the sperm, certain enzymes are released into the ooplasm. అంటే స్పర్మ్ ఎక్రోజోమ్ లో ఉండే సర్టెన్ ఎంజైమ్స్ అనేవి, ఓకే, మీన్స్ ఫెర్టిలిన్ ఎంజైమ్స్ లైక్ దట్, ఇవి ఊప్లాజం లోకి రిలీజ్ అవ్వడం వలన ఈ ఊప్లాజం లో ఉండే న్యూక్లియస్ అనేది స్టిమ్యులేట్ అవుద్ది. అలా స్టిమ్యులేట్ అయినప్పుడు ఈ న్యూక్లియస్ ఆఫ్ ద సెకండ్ oocyte ఏం చేస్తుంది అంటే మియాసిస్ 2ని కంప్లీట్ చేస్తుంది. ఆ మియాసిస్ 2 కూడా ఏంటది, ఏ మియాసిస్. సో ఇది, okay, it is complete the meiosis 2. In this meiosis 2 process, one is, it is the, again, it is a meiosis. Okay, it is also a meiosis. And here meiosis 2 and prophase 2 are completed. Meiosis 2 and prophase 2 are completed here. After that, okay, after that what happened? Here two unequal daughter cells are formed. In this, one is the, okay, in this one is the, what is second polar body. In this one is the second polar body. And large one is the ovum or ooted. Large one is the ovum or ooted. This is, now the secondary oocyte is not able to fertilize it with the sperm nucleus. That's why it will degenerate after some time. It will degenerate after some time. Now the ooted are big size daughter cell and sperm nucleus. These both are get fused and form the zygote. These both are get fused and form the zygote here. These both are get fused and form the zygote. It is the zygote. Now within the, what is, and as usually surrounding of this, the zona pellucida as usually present. Okay, after that what happened? This secondary oocyte, inside the secondary oocyte, what is single what is nucleus is there. Now this nucleus is present in diploid condition, means 46 chromosomes are there. The presence of 46 chromosomes inside the, what is this secondary oocyte, or this nature and this structure is now called as a zygote. And still surrounding the zygote, zona pellucida present. See, what is, see here 23 chromosomes are there, here 23. 23, 23. Total how many? 46 chromosomes. 23, 23. Total 46 chromosomes are there in this zygote. Total 46 means 22 pairs plus XY or XX means the zygote will develop into male or female depend on the fused sperm. I'll explain that. Okay, already in the spermatogenesis I have clearly explained that in the total 100% of spermatozoa, 50% of sperms are having with X chromosome, 50% of sperms are have Y chromosome. See, in these X and Y chromosomes contain sperms, which sperms are having more motility, that is firstly reach to the secondary oocyte and that can fertilize the spermata or that secondary oocyte. For example, X chromosome contains sperm is what is compared with Y chromosome contains sperm, it is more faster. X chromosome contains sperm is more faster than the Y chromosome contains sperm. Now the X chromosome contains sperm is firstly reach to the secondary oocyte and that can fertilize it with secondary oocyte. And the second point is there, that is even the more vigorous capacity is there in Y chromosome contains sperm, okay, which is not able to dissolving the ovular membranes. So even low motility capacity, what is X chromosome contains sperm, if which is able to dissolving the second what is ovular membranes, which is firstly enter into the what is secondary oocyte ooplasm. So it is very clear. So I'll explain. So this is the fertilization. This process, the process of fusion of two dissimilar gametes, sperm nucleus and ovum nucleus within the ooplasm of the secondary oocyte. That process is called fertilization. It is the fertilization. Very clear. It is the fertilization. Now, after completion of the fertilization, the diploid chromosomes contain zygote is formed here. Now, the zygote will develop into male or female. So we will decide that. We will discuss that. Okay. For example, this is the, it is one sperm. Okay, it is one sperm. For example, it is X chromosome contains sperm. It is Y chromosome contains sperm. Okay. And already we are clearly discussed what is that zygote, what is secondary oocyte. The secondary oocyte, it is always contain the X chromosome only. It is always contain the X chromosome. It is always contain the X chromosome. Okay, so it is ovum and it has 22 plus X chromosomes. And this sperms, one sperm, it is with 22 plus X chromosome and here 22 plus Y chromosome. This is the nature. For example, if this sperm is, if these Y chromosome contains sperm, if it is fertilized with, okay, what is this zygote or this ovum, here 22 plus 22, 44, 44 plus here X and Y. Okay, so XY chromosome condition is there in human male. Understood? For example, next, for example, if X chromosome contains sperm, fertile with ovum. See, here 22, here 22. 22 plus 22, what is 22? It is 22 plus 24 to 44 plus here X, here X, X, X. These X, two X chromosome contain in this diploid condition. If two allosomes are, two X chromosomes are allosomes, this zygote will develop into the female fetus. Means the sex determination is depend on the human male. How can we are decide the sex of the fetus will be depend on the human male? Means in which organism the heterozygous chromosomes are present, the sex determination is present on that animal. ఇప్పుడు హ్యూమన్ బీయింగ్ లో XY హెట్రోజైగస్ అంటే 2 డిఫరెంట్ అల్లోజోమ్స్ ఉంటాయి. అదే హ్యూమన్ ఫిమేల్ లో 2 X క్రోమోజోమ్సే ఉంటాయి. అంటే హోమోజైగస్ అల్లోజోమ్స్ ఉంటాయి. అంటే హోమోజైగస్ పేరెంట్ మీద సెక్స్ డిటర్మినేషన్ ఉండదు. హెటరోజైగస్ పేరెంట్ మాత్రమే సెక్స్ డిటర్మినేషన్ లో ఇన్వాల్వ్ అవుద్ది. డిపెండ్ ఆన్ ఫర్టిలైజ్డ్ స్పర్మ్, నాట్ డిపెండ్ ఆన్ ఫర్టిలైజ్డ్ ఓవం. ఎందుకు డిపెండ్ ఆన్ ఫర్టిలైజ్డ్ స్పర్మ్ అంటున్నామంటే, ఇప్పుడు ఎక్స్ క్రోమోజోమ్ కంటైన్ స్పర్మ్ అనేది ఈ ఓవం ని ఫెర్టిలైజ్ చేస్తే ఇక్కడ 2X అంటే ఇక్కడ ఫిమేల్ ఫామ్ అవుతుంది. వాట్ ఈస్ ద క్యారియోటైప్ ఆఫ్ ద హ్యూమన్ ఫిమేల్? 44 ప్లస్ XX, ఇది ఫిమేల్. ఓకే, అండ్ 44 ప్లస్ XY ఇది మేల్. ఓకే, ఇది క్యారియోటైప్. అంటే ఇక్కడ హోమోజైగస్ క్రోమోజోమ్స్ ఉన్నాయి, ఇక్కడ హెటరోజైగస్ క్రోమోజోమ్స్ ఉన్నాయి. అంటే జనరల్ గా ఇంకొక విషయం కూడా ఉంది. సొసైటీలో ఏం జరుగుతుందంటే, ఒక ఉమెన్ కంటిన్యూగా ఫస్ట్ ఆఫ్ స్ప్రింగ్ అయినా ఫిమేల్, సెకండ్ ఆఫ్ స్ప్రింగ్ అయినా ఫిమేల్, ఇలా కంటిన్యూగా అమ్మాయిలే పుడుతున్నారు అనుకోండి, సొసైటీ మొత్తం కూడా ఏమంటుందంటే అది ఆ ఉమెన్ ప్రాబ్లం వల్లనే, ఆ మదర్ వల్లనే అందరు అమ్మాయిలు పుడుతున్నారు ఆమెకి అనేది ఒక కాన్సెప్ట్ డిస్కస్ చేస్తారు. యాక్చువల్ గా ఆ ఉమెన్ రోల్ అనేది జీరో. ఇప్పుడు అమ్మాయిలు పుడుతున్నారు, అబ్బాయిలు పుడుతున్నారు అనే దానిలలో మేల్ స్పర్మే మేజర్ రోల్. అసలు ఓవం రోలే లేదు అక్కడ సెక్స్ లో హ్యూమన్ బీయింగ్స్. ఎందుకంటే ఫిమేల్ నుంచి రిలీజ్ అయిన ఓవం 100% ఓవము ఓన్లీ ఎక్స్ క్రోమోజోమే ఉంటది. బట్ మేల్ నుంచి రిలీజ్ అయిన 100% స్పర్మ్స్ లో 50% X ఉంటుంది, 50% Y ఉంటుంది. ఈ రెండిట్లో ఏది ఎక్కువ యాక్టివ్ ఉంటే అదే ఫస్ట్ రీచ్ అవుతుంది. అది ఫస్ట్ రీచ్ అయినప్పుడు ఏమవుతుంది, అదే ఫెర్టిలైజ్ జరుగుతుంది. ఇప్పుడు Y క్రోమోజోమ్ కంటైన్ స్పర్మ్ కంటే X క్రోమోజోమ్ కంటైన్ స్పర్మ్ ఎక్కువ ఫాస్ట్ విగరస్ స్పీడ్ ఉంది అనుకోండి, ఇదే ఫస్ట్ దీన్ని రీచ్ అయ్యి, దీనికంటే ముందు ఇది రీచ్ అయ్యి దీన్ని ఫెర్టిలైజ్ చేస్తుంది. అప్పుడు జైగోట్ దేనిగా డెవలప్ అవుతుంది, ఇక్కడ XX అంటే ఏది, ఫిమేల్ డెవలప్ అవుతుంది. ఇప్పుడు ఈ ఫిమేల్ ఆఫ్స్ప్రింగ్ ప్రొడ్యూస్ చేసినది ప్రొడ్యూస్ అయింది, ఇక్కడ ఫిమేల్ జైగోట్ ఫామ్ అవుతుంది. ఫిమేల్ జైగోట్ ఫామ్ అవుతుంది అంటే అది ఓవం సైడ్ ఉన్న ప్రాబ్లం, ఓవం రోల్ ఉందా లేకపోతే స్పర్మ్ రోల్ ఉందా? స్పర్మ్ రోల్ ఉంది. స్పర్మ్స్ ఎవరి నుంచి వస్తాయి, హ్యూమన్ మేల్ నుంచి వస్తుంది. అది మేల్ లో ఉండే స్పర్మ్ని బట్టి ఉంటుంది, కానీ ఫిమేల్ లో ఉండే ఓవం బట్టి ఆ జైగోట్ మేల్ గా డెవలప్ కావాలా ఫిమేల్ గా డెవలప్ కావాలా అనేది డిసైడ్ కాదు. అందుకే సొసైటీలో ఇట్లాంటివి చాలా జరుగుతున్నాయి. ఒక లేడీ కంటిన్యూగా అంటే ఇద్దరిని ముగ్గురిని అమ్మాయిల్ని కంటుంది అంటే దాన్ని ఆ ఉమెన్ ని బ్లేమ్ చేస్తున్నారు. యాక్చువల్లీ దట్ ఈస్ అబ్సల్యూట్లీ రాంగ్. అండర్స్టూడ్? దట్ ఈస్ ద ప్రాబ్లం. that is, I don't consider that is the problem. That is not a problem. That is depend on the sperm condition. అది ప్రాబ్లం అనరు దాన్ని. స్పర్మ్ యాక్టివేషన్ బట్టి ఉంటుంది. ఇప్పుడు ఎక్స్ క్రోమోజోమ్ స్పర్మ్ యాక్టివ్ ఉందా, వై క్రోమోజోమ్ స్పర్మ్ యాక్టివ్ ఉందా అనేది డిసైడ్ అవుతుంది. ఫర్ ఎగ్జాంపుల్, ఒక కపుల్స్ ఉంటారు, వాళ్ళకి ఫస్ట్ ఆఫ్ స్ప్రింగ్ మేలే వస్తుంది, సెకండ్ ఆఫ్ స్ప్రింగ్ మేలే, లేదా అమ్మాయిల కోసం ఉంచుకుంటే థర్డ్ ఆఫ్ స్ప్రింగ్ కూడా మేలే ఉంటుంది. అంటే ఏంటి, ఆ హస్బెండ్ లో ఉండే స్పర్మ్ లో వై క్రోమోజోమ్ ఉన్న స్పర్మ్ ఎక్స్ క్రోమోజోమ్ ఉన్న స్పర్మ్ కంటే ర్యాపిడ్ మోర్ విగరస్ స్పీడ్ గా ఉంటది. అందుకే ఫస్ట్ వై క్రోమోజోమ్ అనేది ఎప్పటికి రీచ్ అవుతా ఉంటది. దానివల్ల మేల్సే ప్రొడ్యూస్ అవుతుంటారు. ఇప్పుడు కొంతమందికి కంటిన్యూగా ముగ్గురు అమ్మాయిలు, నలుగురు అమ్మాయిలు అలా. ఇప్పుడు ఫస్ట్ ఒక అమ్మాయి పుట్టింది, అబ్బాయి కోసం మళ్ళా అమ్మాయి పుడుతుంది, అబ్బాయి కోసం మళ్ళా అమ్మాయి పుడుతుంది. ఇలా కంటిన్యూగా అమ్మాయిలు పుడుతున్నారంటే ప్రాబ్లం మదర్ సైడ్ కాదు అక్కడ ఉంది, అది స్పర్మ్ మొటిలిటీ మీద, స్పర్మ్ స్పీడ్ మీద డిపెండ్ అవుతుంది. ఏ స్పర్మ్ ముందు ఫెర్టిలైజ్ చేస్తుందో ఆ జైగోట్ ఈ స్పర్మ్ ఫెర్టిలైజ్డ్ స్పర్మ్ మీదనే డిపెండ్ అవుతుంది. అండర్స్టూడ్? సో at least from this time you should stop and you give the awareness to the society people. So some number of times that women are blamed by the oftenly conceive the female offspring. Okay, so that conceive that what zygote is developed inside the uterus, that is depend on the fertilized sperm, not depend on the fertilized ovum. Understood? Okay, so then our society should aware on this on this what is point, and as a educated person we should have the responsibility for giving the awareness on this. Okay, So by this process, the females are produced. By this process, males are produced. So otherwise, there is no zero role. That woman role is zero for the development of what is male zygote, female zygote. That 100% role is only male sperms. ఓకే, సో ఇది మనం దీంట్లో డిస్కస్ చేసేది. సో దీన్ని బేస్ చేసుకొని ఆ జైగోట్ అనేది మేల్ గా డెవలప్ కావాలా, ఫిమేల్ గా డెవలప్ కావాలా అనేది డిసైడ్ అవుతూ ఉంటది. ఇది అప్ టు హియర్ ఇట్ ఈస్ ద వాట్ ఈస్ జైగోట్ ఫార్మేషన్. ఓకే, ఆఫ్టర్ కంప్లీషన్ ఆఫ్ ద జైగోట్, ఓకే, నౌ విల్ గో టు ద వాట్ ఈస్ fallopian tube. Where does the fertilization takes place? In the ampullary region. That is clear. Where does the fertilization takes place? Means in the ampullary region. That is clear, right? After this, okay, for example, it is the zygote. Okay, surrounding the zygote, it is the single cell. Zygote, single cell, 44 plus XY. 44 plus XY. And still surrounding of this zona pellucida is still present. Zona pellucida present. Still this total structure is present in the ampullar region. This entire structure is present in the ampullar region it is. Okay, this total structure is okay. So this is the what is zygote. Okay, surrounding the zygote still zona pellucida present. I'll write I'll give another color for the zona pellucida. This is the zona pellucida. Okay, now it is the zygote. So after completion of the fertilization, the zygote move from ampulla to isthmus, isthmus to that is enter into the uterus fluid. That is enter into the uterus fluid. Now the zygote after 30 hours, okay, after 30 hours, after 30 hours here, after 30 hours, okay, so what is the zygote is started the cleavage process, and sometime here called after 36 hours are also. Okay, now within the zygote the first cleavages are started. So within the first cleavage, what is the which structure has major role in the what is completion of first cleavage within the zygote? Okay, so it is cleavage. It is cleavage. See, for the completion of first cleavage within the zygote, what is the proximal centriole has major role in this. Proximal centriole. Proximal centriole has major role in this. So already we have discussed. Along with the nucleus, the proximal centriole is also released into the ooplasm. So after 30 or 36 hours later from the fertilization, this proximal centriole can initiate the first mitotic division within the zygote. After completion of the first cleavage, see, in the embryonic development, we will use some points, some words here. Please note it down. One word is the embryo, and second one word is the fetus. Okay, so what is the fetus? Which age of the gestation period is called fetus? Which age of the gestation period is called embryo? Okay, so in the gestation period first to 8 weeks, first week to 8 weeks of gestation period, 8 weeks of gestation, 8 weeks of gestation period, 8 weeks of gestation period. Gestation period, I'll use GP word. And what is 9th week to, okay, 9th week to end of the gestation period. End of the gestation period is called the fetus. From first day of the fertilization to 8 weeks of the gestation period is called embryo, and beginning of 9th week to end of the gestation period is called fetus. And one more important point is there here. That point is, in embryonic stage, the daughter cells are called blastomeres. The daughter cells are called blastomeres. Blastomeres are called here daughter cells. Daughter. These are the daughter cells. And here, as usually, it is called as a daughter cells. As usually, here called as a daughter cell. There is no another name. These are called as a daughter cells. Blastomeres, daughter cells. And in the embryo, the mitotic divisions are called cleavage. Cleavage. Okay, here, what is the another name of mitotic divisions? Cleavage. Mitotic process or mitotic divisions. Mitotic divisions are called cleavage, and here as usually called as a mitotic divisions. As usually called as a mitotic divisions. So it is very clear information. It is very clear. So in embryonic development, that mitotic divisions are called cleavage, and daughter cells are called blastomeres. Right? Here, as usually daughter cells. Okay. See, after completion of the first cleavage in the zygote after 36 hours, and here how many daughter cells? Nothing but how many blastomeres? 2 blastomeres are developed here. 2 blastomeres. Okay. See, again after this, again one more cleavage completed here. Okay. Here second cleavage is completed. Second cleavage. After completion of the second cleavage, what happened? So again 4 blastomere cells are formed here. 4 blastomeres. These are the 4 blastomeres. 4 blastomere cells. 4 blastomere. Again and again in this process, by the third, how many? 8. See, it is the zygote. Okay. After within the what is zygote? Right. So it is a single cell. Okay. So after first cleavage, 2 blastomeres, and after second cleavage, what is 4 blastomeres, 4 blastomeres, and after third cleavage, 8 blastomere, after fourth, 16 blastomere, and after fifth, that is 32 blastomere, after sixth cleavage, 64 blastomere, after seventh cleavage, 128. Like that, in this process continuously, in every cleavage process, that number of blastomeres are becomes double. Number of blastomeres are becomes double in every cleavage. In every cleavage process, the blastomere cells are becomes double. So in this process, whenever the zygote is reached to 8 to 16 cell stage, 8 to 16 blastomeres. Okay, 8 to 16 blastomere, and in this condition, it is look like a oval shape, and it is look like a mulberry shape. That's why the name of this stage is called morula. It is the morula stage. These are the embryonic stages. This is the first stage of the embryo. Okay, it is the first stage. It is the first stage of embryo. Understood? Right. After that, by the repeated mitotic divisions, what happened? Continuously by the repeated mitotic divisions continuously, what happened? The number of daughter cells are increased by the increasing the number of daughter cells here. Okay, so what is, whenever these embryos blastomeres are what is cross more than 32 blastomeres. Okay, in this condition, that stage is called as a blastula. It is completely round shape. It is oval shape. It is round shape. This stage is the blastula. That stage is the blastula stage. Okay, that stage is the blastula stage. Right. After that, this blastula stage is directly enter into the this blastula stage directly enter into the uterus fluid through the isthmus region. Through the isthmus portion, what is this blastula is enter into the what is uterus region. Uterus for what purpose? Implantation purpose. For the implantation purpose, this what is zygote or embryo is enter into the uterus fluid. It is enter into the uterus fluid, and I'll explain with another picture here. See, for example, what is it is the fallopian tube. Okay, it is the uterus lining. It is the uterus lining, and this is the endometrium. It is the endometrium. It is clear, right? After the it is the uterine fundus. Okay, it is uterine fundus. Now, what is the whenever the blastula stage enter into the uterus fluid, it is the blastula stage. The blastula stage is enter into the uterus fluid. Okay, it is the blastula. Blastula. It is the blastula. The blastula is enter into the uterus fluid. After entry of this blastula into the uterus fluid, now this is the uterus. It is the uterus. Okay, so when the blastula enter into the uterus fluid, it is undergoes to cavitation. It is undergoes to cavitation. See, capacitation is one word is there. Okay, cavitation is one word is there. These are start with the C. These are start with which letter? C letter. These are start with the C letter. Okay, cavitation means what? See, the cavitation process is done by the one process, that is the compaction. That is the compaction. So what is the meaning for compaction? Okay, compaction is one kind of cell adjustment process. Okay, compaction is kind of cell adjustment. అంటే ఇక్కడ సెల్ అడ్జస్ట్మెంట్ ప్రాసెస్ అంటే ఇప్పుడు ఇది బ్లాస్టులా స్టేజ్. ఇది ఈ బ్లాస్టులా స్టేజ్ లో సెల్స్ అనేవి గ్యాప్ జంక్షన్స్ ఉంటాయి. బిట్వీన్ ద సెల్స్ పర్టికులర్ గ్యాప్ ఈస్ దేర్. ఓకే, దట్స్ వై దట్ ఈస్ కాల్డ్ గ్యాప్ జంక్షన్. సో ఈ గ్యాప్ జంక్షన్స్ ఉన్న బ్లాస్టులాలో కంపాక్షన్ అనే ఒక ప్రాసెస్ జరిగినప్పుడు ఏమవుతుందంటే ఈ బ్లాస్టులలో ఉండే సెల్స్ అనేవి టోటల్ 2 గ్రూప్స్ గా డివైడ్ అయిపోతాయి. One group of cells are arranged in outer layer. Okay, that outer layer cells are called here tropoblast. Okay, దీంట్లో one group of cells అనేవి ఫామ్ అవుతాయి. ఇవి ఒక లేయర్ గా అవుటర్ లేయర్ గా ఫామ్ అవుతాయి. ఈ అవుటర్ లేయర్ ఆఫ్ దీస్ బ్లాస్టోమియర్ సెల్స్ ఆర్ కాల్డ్ ట్రోపోబ్లాస్ట్. అండ్ సెకండ్ గ్రూప్ ఆఫ్ సెల్స్ ఫామ్ అవుతాయి ఇక్కడ. సో దీని ఇన్సైడ్ సెల్ అన్నిట్లలో ఈ గ్యాప్ అనేది డిక్రీజ్ అయిపోద్ది. సో వన్ సెల్ ఈస్ టైట్లీ what is attached to another. So these are developed as a one more type of cells. These are called here inner cell mass. ikkada idi very important point, ikkada very clear. అంటే ఇక్కడ బ్లాస్టులా స్టేజ్ లో ఉండే ఈ టోటల్ బ్లాస్టోమియర్స్ 2 గ్రూప్ ఆఫ్ సెల్ గా డివైడ్ అవుతాయి ఈ కంపాక్షన్ వల్ల. ఈ కంపాక్షన్ లో వన్ గ్రూప్ ఆఫ్ సెల్స్ ఏమో అవుటర్ లేయర్ లో ట్రోపోబ్లాస్ట్ ని ఫామ్ చేస్తాయి. నెక్స్ట్ ఇన్సైడ్ సెల్స్ ఏమో ఈ గ్యాప్ అంతా అడ్జస్ట్ అయ్యి స్పేస్ మొత్తం తగ్గిపోయి అంటే ఒకదానికి ఒకటి టైట్ గా అటాచ్ అవుతాయి. ఇలా టైట్ గా అటాచ్ అవడం వల్ల ఇన్సైడ్ బ్లాస్టాలో ఒక ఎంటీ స్పేస్ ఫామ్ అవుతుంది. ఈ ఎంటీ స్పేస్ ని మనము సిస్ట్ అంటాం లేదా క్యావిటీ అంటాం. సో విదిన్ ద blastula stage the cavity or cyst is formed. So this process is called cavitation. Capacitation, compaction, cavitation. These two C's are there, 3C. Okay. So after that, this stage is called blastocyst. After that, this stage is called blastocyst. It is one, and within this blastocyst, still surrounding of the blastocyst also, what is the zona pellucida still present. Okay, zona pellucida still present the surrounding the blastocyst. This is the cavitation. See here, one points here. Capacitation is one point. Capacitation, it is one point. Next, what is compaction is one point, and cavitation is one point. Cavitation. Okay, these are a few points here. Very important thing. And after completion of the blastocyst, within the uterus fluid, certain enzymes are there. Within the uterus fluid, certain enzymes are there. These enzymes are get dissolving the zona pellucida. Now the zona pellucida get dissolved by the action of certain enzymes within the uterus fluid. Okay. After that, what happened? Zona pellucida get dissolved. Okay. Zona pellucida. Right, it is. Zona pellucida get dissolved. Get dissolved by the action of uterus fluid enzymes. By the action of uterus fluid enzymes. Uterus fluid contain enzymes. By the action of uterus fluid contain enzyme. After that, what happened? So the uterus means zona pellucida can dissolve. Zona pellucida can dissolve. After dissolving the zona pellucida, what happened? See, this blastocyst is attracted by the endometrium of the uterus. Okay, it is the endometrium. Now the endometrium attracting to the blastocyst stage. ఇక్కడ ఇంకొక పాయింట్ ఉంటది. Which stage of the embryo get attached with endometrium? Okay, that stage of the embryo is the blastocyst. That is clear. That is called implantation. That is called what? Implantation. The process of attachment of this blastocyst with the endometrium is called implantation. I'll show this picture again. For example, it is the lining of endometrium. It is the lining. It is a lining of endometrium, and it is the endometrium. After that, what is this embryo is means blastocyst is attached to the endometrium, and again inner cell mass is also there inside the blastocyst, and again the endometrium is there. This is okay. This process is called implantation. Implantation. The process of attachment of the blastocyst with endometrium is called implantation. It is very clear important point, that is blastocyst. This is the blastocyst. The process of attachment of blastocyst with endometrium is called implantation. It is very clear, right? After completion, see at which day from the fertilization the implantation process takes place. So implantation process takes place. It is takes place after 6th day from the fertilization. After 6th day. After 6th day, from the fertilization. After 6th day from the fertilization. So in some reference books given, actually this word is not there in NCERT, but in some reference books given, at the 6th day from the fertilization, implantation takes place, and after the 6th day what is the implantation takes place. But in our academic books, after 6th day word mentioned, that's why we go through that word. And after 6th day the blastocyst get attached with endometrium. After attachment of this endometrium, what is this blastocyst with endometrium, gradually the endometrium is overlapping the implanted embryo. Now the total embryo is embedded within the endometrium, means the total embryo, means that blastocyst is completely overlapping by the endometrium. So thereby what happened, the blastocyst get support, the blastocyst get more support from the endometrium. Okay, the blastocyst get more support from the endometrium here. It is very, very clear, important point. Okay, after that, next go to the pregnancy and embryonic development. Okay, now we will start. Okay, right. See, after it is the implantation process. See, after completion of the implantation, for the implantation, which hormone has major role? Which hormone has major role in the implantation? So for the implantation, okay, for the implantation, the progesterone hormone has major role. For the implantation, progesterone hormone has major role. So the progesterone hormone, it is involved in the development of endometrium. Okay, progesterone hormone has major role in the development of endometrium. Okay, and here few questions are there here. So what is the reason some women are after conceive, they are suffered by the oftenly abortion? Okay, కొంతమంది ఉమెన్స్ కన్సీవ్ అయిన తర్వాత, ఓకే, సో సెకండ్ మంత్, థర్డ్ మంత్, ఫోర్త్ మంత్లో ఏమైందంటే అబార్షన్ అవుద్ది. అలా అబార్షన్ కావడానికి రీజన్ ఏంటి అనేది ఒక పాయింట్ హియర్. ఇక్కడ ఒక పాయింట్ డిస్కస్ చేయాలి. వాట్ ఈస్ ద రీజన్ ఫర్ అబార్షన్ టేక్స్ ప్లేస్? Reason is the deficiency of this progesterone hormone. So some questions are there here. That questions are, okay, so one woman oftenly suffered by the abortion. When that woman consult the gynec doctor, the gynecologist suggests the one hormone contain pills. Now that pills are contain which hormone given below? Understood? That hormone is the progesterone hormone. That hormone is what? Progesterone hormone. It is very clear. Progesterone hormone, they will suggest. What is the role of progesterone hormone? Means the progesterone hormone, it is helps to development of endometrium by the increasing the concentration of endometrium, that is giving more support to the implanted embryo. That is giving more support. For example, after implantation the endometrium is not developed, so and by the increasing the number of cells, that gradually the weight of the fetal embryo is increased. By the increasing the weight, gradually the implanted embryo is gradually it is get detached. After detaching, what happen? This goes to abortion. అంటే ఇక్కడ ఇది స్ట్రాంగ్ ఎంబ్రియో అనేది ఎండోమెట్రియం తోటి స్ట్రాంగ్ అటాచ్మెంట్ ఉండాలి అని అంటే ఎండోమెట్రియం డెవలప్మెంట్ ఎక్కువగా ఉంటేనే ఎంబ్రియోని మొత్తం ఇలా ఓవర్లాప్ చేస్తుంది. ఇలా ఓవర్లాప్ చేసినప్పుడు ఈ ఎంబ్రియో అనేది స్ట్రాంగ్ సపోర్ట్ ఉంటుంది ఎంబ్రియోకు ఎండోమెట్రియం నుంచి. సో ఇలా ఇప్పుడు ఇదంతా ఎండోమెట్రియం రెడ్ కలర్. ఓకే, ఇది దీన్ని మొత్తం ఓవర్లాప్ చేయడం వల్ల ఈ ఎంబ్రియో అనేది స్ట్రాంగ్ గా సపోర్ట్ తీసుకుంటుంది ఎండోమెట్రియం నుంచి. లేదు అంటే ఇది డెవలప్ కాలేదంటే దీని వెయిట్ పెరిగే కొద్దీ ఇది వెయిట్ పెరిగి గ్రాడ్యువల్ దీని నుంచి డిఅటాచ్ అవుతుంది ఎంబ్రియో. ఇది డిఅటాచ్ అయిపోయినప్పుడు ఏమవుతుంది? అబార్షన్. అంటే ఇది సర్విక్స్ లోకి వచ్చి సర్విక్స్ నుంచి వెజైనా నుంచి అబార్షన్ జరుగుతుంది. ఓకే, మరి అబార్షన్ జరగకుండా ఏ హార్మోన్ ని సజెస్ట్ చేస్తారంటే గైనకాలజిస్ట్ ప్రొజెస్టరాన్ హార్మోన్ కంటైన్ పిల్స్ ని సజెస్ట్ చేస్తారు. Okay, so ఇది ఒక ఇంపార్టెంట్ పాయింట్. ఆఫ్టర్ దిస్, what is the reason after pregnancy? Okay, after pregnancy, why the menstrual cycle will be stopped? Okay, it is very important point. It is after conceive, why the menstrual cycle will be ceased, or why the menstrual cycle will be stopped? Why the menstrual cycle will be stopped? Why the menstrual cycle will be stopped? That's enough. Okay, so reason, there is one reason is there. Once the sperma fertilized with ovum, the corpus luteum lifespan will be increased. For example, after ovulation, if the sperm is not fertilized with ovum, what happened? The corpus luteum lifespan is only 14 days. Okay, అంటే స్పర్మ్ ఓవులేషన్ తర్వాత స్పర్మ్ ఓవుని ఫెర్టిలైజ్ చేస్తే, ఓకే, ఫెర్టిలైజ్ చేయకపోతే, ఫస్ట్ నేను ఫెర్టిలైజ్ చేయకపోతే ఏం జరుగుతుంది, ఫెర్టిలైజ్ అయితే ఏం జరుగుతుంది అనేది చెప్తా. కార్పస్ లుటియం లైఫ్ స్పాన్ ఈస్ డిపెండ్ ఆన్ ద ఫెర్టిలైజ్డ్ ఓవం. ఓకే, కార్పస్ లుటియం లైఫ్ స్పాన్, కార్పస్ luteum lifespan, corpus luteum lifespan. Okay, it is depend on fertilizing ovum, depend on fertilized ovum. Okay, if, very important point, if ovum not fertilized with sperm, ovum not fertilized with sperm, okay, lifespan of what is corpus luteum. Okay, lifespan of corpus luteum is 14 days only. 14 days only. Okay, if the sperm fertilized with ovum, if ovum fertilized with sperm, if ovum fertilized with sperm, fertilized with sperm, lifespan of corpus luteum, is 3 to 4 months. 3 to 4 months. So what is the reason the lifespans of corpus luteum will be changed oftenly? Oftenly means depend on fertilized, non-fertilized. Why the, what is these lifespans are will be changed? Reason is there. See, if to the ovum the sperm is not available, there is no fertilization. If the fertilization is not takes place after ovulation, the empty graphene follicle is called corpus luteum. Okay, so the corpus luteum is involved in the production of estrogen, progesterone. In yesterday class we are discussed, means in previous class we are discussed that point. Okay, corpus luteum can produce estrogen and progesterone hormones. Okay, so if to the ovum sperm is available, the corpus luteum lifespan will be prolonged. Means in these 3 to 4 months, why the 3 to 4 months corpus luteum lifespan is there? Means within these, up to these 3 to 4 months, the placenta development will be takes place. The placenta development will be takes place here. Okay, placenta development here. So this is the placenta development is takes place, and in this 3 to 4 months, as usually, estrogen and progesterone hormones are continuously produced. Now the presence of this corpus luteum, see, this corpus luteum is also maintained by the luteinizing hormone, and that one pregnant hormone is there. What is corticotrophic releasing hormone is there, that is. Okay, So these two hormones are maintain the, what is this, corpus luteum. Corpus luteum, that is the gonadotrophic releasing hormone, that is gonadotrophic releasing hormone. And it is also called with human choriogonadotrophin, that is human choriogonadotrophin hormone, that is HCG. These two hormones are maintained the corpus luteum. By the maintenance of corpus luteum, the corpus luteum continuously produces the progesterone hormone. The continuous production of progesterone hormone takes place. This progesterone hormone inhibit the, this progesterone hormone inhibit, this progesterone hormone inhibit the, okay, inhibit the production of LH and FSH, production of LH and FSH hormones, LH and FSH hormones. So without these gonadotrophins, there is no menstrual cycle. Without these two hormones, okay, next menstrual cycle will not continue. There is no next menstrual cycle. Okay, so next menstrual cycle absent, no next menstrual cycle. So these are the important points regarding these corpus luteum and these explanation. Okay, so this is the important information regarding this. Okay, so after this, next we'll continues to the pregnancy and embryonic development, including we will discuss some animals gestation periods. Okay, and then we'll complete the what is a complete embryonic development in the next session. Until that, you keep watch these videos and these classes, and if you have any doubts in these classes, just you type your message in what is in comments role, then we'll give the answers to you, right? We'll meet in the next session, in the next class. Okay, thank you.